All right, so we're going to start off today by talking about just the real basics of what you're going to need to know for the rest of the class. And a lot of this is going to be review for you. If it's like super old school review, you're not needing to go over this again, then the PowerPoint for this is in your doc sharing, and you can kind of pop through that and make sure that you're comfortable with all the information. But if you want to have an audio in addition to the video, not really video, but the slides that we're going through here, this is a nice way for us to kind of go over this information relatively quickly. This should be a short little video here. So this is the basics of cell biology. So we're going to talk about the individual cell and why it's important for us to know about that in order for us to learn more specifically about the different types of cells that we're going to talk about, in particular how they refer to hematology. So let's get started. So cells are our basic unit of all living things. We have our single-celled organisms, also called unicellular. These are oftentimes what we see in our fecal samples as parasites. Now the organisms themselves, the parasites, are not always single-celled. There are some that are, like Coccidia and Giardia, for instance, are single-celled organisms. But then we will also see the ova, or the eggs of those organisms, as single cells when in a fecal sample as well. So that's kind of a little analogy we can use there. And then most of the organisms we're going to be studying, especially in this class, are going to be complex organisms, so multicellular organisms, like our canines and felines and us, and they're basically made up of lots and lots of different types of cells. So we're going to be looking at, in particular, the hematologic or blood cells in this class. The cells are all microscopic. You guys know this already. We typically use microns or micrometers as the unit that we're going to measure these cells in. And the average cell is 10 to 30 microns. When we look at red blood cells, however, we're looking at smaller than that. Typically for our dog and cat, we're looking between 5 to 8 microns, so a little smaller than that, but that's just our average here. And that we will use a microscopic scale. Many microscopes have a little scale on there that you can measure the size of the cell. So that might help to bring it into a little bit more clarity as you're looking at these cells underneath the microscope. But remembering that red blood cells of dogs and cats are between 5 and 8 microns might give you a little bit of reference point when you're looking inside the microscope at different cells. Now, the type of cells I'm going to mostly talk about today are our typical complex cells that make up the majority of our body parts. And these are going to refer more specifically to the white blood cells that we're going to be studying in this class. Red blood cells are a little bit different, especially mature red blood cells, and we'll talk a little bit more about that in a minute. But all of the cells, no matter which ones we're talking about, are going to have an outside membrane, a cell membrane. This is also called a plasma membrane. And this is sort of like, if you think about a cell as sort of like a bag of jelly, this is the outside bag. This is the outside of the Ziploc bag that's holding everything together. It's creating a barrier between the extracellular or interstitial tissue or space and the inside environment of the actual cell. And there's going to be transport of nutrients and electrolytes and action potentials if we're talking about nerve cells. This is all going to be regulated by that outside lining or outside membrane of the cell that we're looking at. And in this particular image, it's that purple plasma cell membrane. So that's what we're looking at, is the outer portion of the cell. It gives the cell its structure, and very often it gives the cell its definition and helps us to identify based on the type of the outside membrane allows the cell to create a shape and a particular structure that allows it to be more identifiable. So the next part of the cell we're going to talk about is the nucleus, and that's in that center of this picture. It's kind of pink with little yellow circles on it in this particular image. And all of our white blood cells are going to have nuclei, which is the plural for nucleus. And the difference with our red blood cells is that a mature red blood cell will not have a nucleus. That will be pushed out of the cell as the cell matures. But when we do the maturation stages, we're going to be talking about the nucleus as it refers to the ribosome cell as well, or the red blood cell. So what does a nucleus do? It's part of the organ system of the cell, and we call cell organs organelles because they're little, and they're not exactly full organs like you think of in a mammal. And this, we kind of refer to this part of the cell as the brain of the cell. And the reason why we refer to it as the brain is that it controls most of the activities in the cell. It also is going to facilitate or regulate cell division, or how that cell divides into more cells. The nuclear membrane keeps the nucleus all in one area, and that controls the movement of molecules, nutrients, different structures in and out of the nucleus as well. And so we have a plasma cell membrane, then we have a nuclear membrane that creates the structure for the nucleus. And then as we start to look at our different white blood cells, you'll notice that we have really different structures of the nuclei depending on the cell. And again, that creates another recognizable component on how we can identify the different cells from each other. Inside the nucleus, oftentimes will appear this little circular dot, and in the 2D image of a slide, we'll see this darker staining, typically, dot within the nuclear membrane, within the nucleus itself. These are usually round, and there's usually one, but there can be multiple, and especially in a cell that's getting ready to divide or that has a really rapid division program kind of programmed into it, we'll see more than one nucleolus, and those are called nucleoli or nucleoli. It depends on how you want to pronounce that. These contain ribosomes and act as a reservoir for RNA that helps to regulate cell division and cell metabolic processes. RNA and protein are going to make up this this nucleoli structure within the nucleus as well. Chromatin is within the nucleus, and this is what's containing our DNA, the genetic information of each individual cell. This is going to be usually kind of loose within the nucleus until it's time for that cell to divide, and it's containing all the information that's necessary for the cell to create proteins and for the different types of activity that that cell will undergo, and that's oftentimes dependent on what kind of cell it is. Once the cell is ready to divide, it's actually that chromatin is going to start to compact itself, and it's going to basically condense into these little X-shaped structures called chromosomes. And they're going to line up along the center line of the cell, duplicate themselves, and then divide in the case of cell mitosis, which we'll look at an image at the end of this little PowerPoint where you can see that. Now, inside the plasma cell membrane, where all these little organelles are floating, is this kind of gelatinous fluid type material called cytoplasm. It's a little bit sticky, so it's got some viscousness to it, and it basically just provides an area for all the organelles to float around in and provide some nutrition and nutrients to them as they are floating around and performing their various functions within the cell. Molecules, different nutrients, electrolytes, proteins are all transferred all around in the cytoplasm into the different organelles they need to go to, in and out of the cell membrane, kind of all around, floating around inside the cytoplasm here. So the fluid is called cytosol. There's also a cytoskeleton, which is kind of the frame for the cell, helps it keep its shape. The organelles, we talked about those. Those are those little cellular organisms like the nucleus and the ribosomes and the mitochondria. And then we also have these little inclusions. And this is especially important in cells that phagocytize or that eat other things in the body. Phagocytic cells will have lots of inclusions that will incorporate all the stuff that the cell has eaten or engulfed. All right, so sometimes we talk about the structure called the powerhouse of the cell, and the actual word for this is the mitochondria. And mitochondria are very, very important. They're going to create the material called ATP, or adenosine triphosphate, that the cell needs in order to function. It's basically going to create the energy for the cell. And the mitochondria have a really specific structure. You can see in this image, they're little represented as yellow in the structure, and there's 4 of them actually, in this image, and they kind of have this like sort of weaving back and forth little picture to them. So the mitochondria are producing energy for the cell, about 95% of the energy that fuels the cells, and that's going to use oxygen to do that. So that's one of the reasons why oxygen is so important to the function of all of the cells in the body. They're also going to help to break down carbohydrates and fats and protein, and then that's going to be stored as ATP. And so some of that carbohydrate is going to come in the form of glucose. We're going to have fats and proteins that are part of that structure as well. And then ATP can be used for lots of different cellular functions as the energy. So certain cells have lots of mitochondria, like heart cells and brain cells, compared to endothelial cells that line the blood vessels. So those don't have a lot of mitochondria because they don't have to do a whole lot. But heart cells are working constantly, brain cells are working constantly. So there's going to be a lot more mitochondria depending on the function of the particular cell. I just like the name Golgi apparatus. It just sounds kind of cool. So Golgi bodies and Golgi apparatus, they look sort of like these layers of pancakes. You can see in this image it's represented by the orange on the right side of the cell, those orange little flattened pancakes. So the purpose of the Golgi apparatus is to store and then create little packages of secretions that the cell is going to get rid of, waste products and other products that the cell is creating within the cell. They're going to package them up so that they can be released into the bloodstream or into extracellular fluid or something of that nature. So it's kind of like the UPS station. They're basically putting things together in packages and shipping them out for either waste product removal or for use in other parts of the body. We also have these little structures called ribosomes, and these are formed by the Golgi apparatus, and these are sort of like the stomach of the cell. They have digestive enzymes that help the protein break down, or excuse me, help the cell break down protein molecules. Anything that's been phagocytized gets broken down by lysosomes, like bacteria or viruses or fungal elements that the cell has phagocytized. So they're basically the enzymatic and stomach digestive function of the cell. So cell division can happen in 2 different ways. We can have meiosis, and this is strictly the gametes or the sex cells like ova and sperm. Meiosis only occurs in the ovaries and in the testes of the mammals that we're going to be studying. And so these cells will divide and have only 1/2 of the genetic material of the parent cell. And that's because we need an ova and a sperm from 2 separate organisms in order to come together to make one full copy of the new organism that is being created with sexual reproduction. Whereas mitosis, this is going to involve growth of basically all of the other cells. Every other cell besides the gametes are going to divide by mitosis. And this is basically replication of the cell creating two identical cells. They're called daughter cells. And so the cell basically duplicates all of its genetic material and then divides into two separate cells. I've got a couple of images here to show you the difference between the two. So this first one is meiosis, and we have a cell on the left with a set of chromosomes, one in red, one in blue. And then we get DNA replication, so we get these chromosomes that duplicate themselves. And then there's some recombination, that's the mixing up of the red and the blue that you see there. Then what happens is as the cells divide, one half of the chromosomes go to one portion of the cell and the other half of the chromosomes go to the other portion of the cell. And then we end up getting two cells, each with one half of the genetic material. And that divides out again until we have one half of the genetic material in each of the gametes, which would be the eggs or the sperm cells. Now in mitosis, it's a little more simple of a process. We have up on the top left, we have prophase, and that's just the cell kind of hanging out as it's supposed to be. And then in prometaphase and metaphase, the chromosomes all duplicate. So we get two copies of the genetic material for the cells. They line up along the center of the nucleus, and they line up in a little line, and then they kind of get pulled to either side of the cell, and the cell divides, stretches, and splits into two. And so then you have in anaphase, everybody's getting pulled off to the side. In telophase, the cells are being divided in two, and then in interphase, everything kind of settles down again until we start prophase to do the division again. In interphase, those cells are basically just like the parent cell. They have the same exact genetic material in each cell. So that's mitosis. And all of the cells that we're going to study in hematology, red and white blood cells, and platelets all divide by mitosis. All right, if you have questions, feel free to ask me in class or send me an email outside of class, and we'll see you in the next one.